Hello everyone, in this video we will learn about what is recombinant DNA technology. That will include the tools such as target DNA, restriction enzyme, cloning vector, host cells, and modifying enzyme. So what is recombinant DNA technology? Recombinant DNA technology is one of the methods in genetic engineering in which we have a direct access to the gene or to the DNA. In other words, we will have a direct manipulation of the gene. We can cut, we can replace specific gene or part of the DNA so that the DNA or the gene able to produce specific product that we want, such as insulin, growth hormone that will be useful in growth hormone therapy, such as to treat growth hormone deficiency in human. Recombinant DNA technology will combine genes from different organisms in vitro or outside of the cell of living organism. Generally, in recombinant DNA technology, we transfer specific target gene into a vector such as plasmid, and then this plasmid will be uptaken by bacteria, and then the bacteria later on able to produce protein that we want. Next, let's learn about the tools that we use in recombinant DNA technology. First, we have the target DNA or gene of interest. Target DNA is a specific DNA segment that contains gene of interest that code for desired protein. This target gene can be extracted from human, animals, and plant. Here is the few examples of target gene. The gene that code for insulin from human, somatotropin gene from cow, gene that code for beta-carotene from the corn. Now let's learn about restriction enzyme. Restriction enzyme, also known as restriction in the nucleus, that can recognize and cut a specific DNA segment. Imagine restriction enzyme like a scissors to cut the DNA. We will learn about two types of restriction enzyme, EcoR1 that will produce sticky ends, and SMA1 that will produce blunt ends. The restriction enzyme extracted from the bacteria, and its natural function is to cut DNA or RNA from the other organisms that enter the cell. The restriction site is the part of the DNA where the restriction enzyme can recognize and cut the DNA. The restriction site, also known as palindromic sequence, in which the base pair are read the same in opposite direction on both complementary strands. The next tool is DNA cloning vector. DNA cloning vector is a DNA molecule that can carry foreign DNA into a host cell and will be replicated inside the host cell. Here is the few types of cloning vector. The most commonly used cloning vector is a plasmid because it is small size that is easy to manipulate. Cloning vector must able to accept foreign DNA on its multiple cloning site, able to replicate freely because it has its own origin of replication, and also possess selectable genetic marker, that is gene that code for antibiotic resistance. Here is the example of cloning vectors such as bacteriophage, cosmid, and the yeast artificial chromosome. Next, we will discuss about the host cell. The host cell is a bacteria that able to accept or receive the recombinant DNA. The bacteria that uptake the recombinant plasmid are said to be transformed. The bacteria that is most commonly used as a host cell is Escherichia coli or E. coli. Bacteria will uptake recombinant plasmid through transformation process to form recombinant bacterium. Now we will discuss about modifying enzyme. DNA ligase is the enzyme that can join the target gene and cloning vector by catalyzing the formation of phosphodiester bond. Imagine that DNA ligase will act as a glue to seal the phosphodiester bond. The next modifying enzyme is TAG polymerase, the thermostable DNA polymerase isolated from bacteria known as Thermus aquaticus. This bacteria grows in the area of hot spring. TAG polymerase normally used to synthesize DNA during polymerase chain reaction. Now we will discuss more about what is restriction enzyme. As we learned earlier, restriction enzyme is the enzyme that recognizes and cuts the specific sequence on the DNA that called restriction site or palindromic sequence to produce sticky ends or blunt ends. The restriction enzyme EcoR1 produce sticky end extracted from the bacteria Escherichia coli. Recognize the restriction site 5' GAA TTC 3'. While for SMA1 that will produce blunt ends extracted from Serratia marcescens, recognize sequence of 5' CCC GGG 3'. The restriction enzyme EcoR1 produce sticky end because the restriction enzyme cut the DNA strands at a different position. While for SMA1, it will produce blunt end because this restriction enzyme cut both strands of the DNA at the same position. The natural function of the restriction enzyme in bacteria is to cut any foreign DNA and RNA that may enter the cell. The bacterial DNA is protected from its own restriction enzyme because of the methylation of the restriction site. Now we will discuss about plasmid as a cloning vector. Plasmid consists of three components: the multiple cloning site, origin of replication, and selectable genetic marker. Plasmid is a small circular double-stranded DNA that is capable of replicating independently within the host cell. Plasmid is the excellent choice as a cloning vector because it is able to accept foreign DNA, able to replicate freely, and possess selectable genetic marker. Now we will discuss about the modifying enzyme, DNA ligase and TAG polymerase. DNA ligase will catalyze the reaction between the phosphate group of the 5' end and the hydroxyl group of the 3' end of the DNA strand. In recombinant DNA technology, the DNA ligase will seal the backbone or the phosphodiester bond between the target gene and then the plasmid to form the recombinant DNA. Next, we will learn about another type modifying enzyme, that is TAG polymerase. TAG polymerase is a heat-resistant enzyme or a thermostable DNA polymerase that is useful in polymerase chain reaction to amplify the fragment of DNA. This is because the TAG polymerase can withstand a very high temperature. PCR or polymerase chain reaction will involve the thermal cycling process. In some steps, the temperature need to increase up to 95 degrees Celsius. The thermostable enzyme take polymerase is useful in this process.